Hello, it's Shridan and today we're going to be solving October-November 2020 Paper 1 Variant 1. Before we start, we are offering free Topic 1 notes. All you have to do is fill in the form in the description box down below. We are also selling Topic 2 notes which include all the tips, tricks of Paper 1 and common misconceptions with worked examples too. Upon purchase, you can ask us unlimited number of questions about the topic or past paper questions totally for free, and you are able to exclusively contact us at any time, which is a new and unique service. Question number 1. Which row shows the correct order of size of these cell structures? Back to our notes, clearly we could see that the chloroplasts have the largest size in a eukaryotic cell, and then the mitochondria is the second largest, then the ribosomes with a width of 13 nanometers, then the cell surface membrane of a length of 7 nanometers in diameter. Therefore, the answer depending on our table is going to be C, with chloroplast being the largest and the cell surface membrane width being the smallest. Question number 2. The diagram shows a stage micrometer scale viewed through an eyepiece containing a graticule. The small divisions of the stage micrometer scale are 0.1 millimeters. The stage micrometer scale is replaced by a slide of a plant cell. What is the actual length of the nucleus of the plant cell? So let's first start by identifying which is the micrometer scale and which one is the eyepiece graticule. Here, this is the stage micrometer, and this ruler divided into 100 divisions is going to be the eyepiece graticule. Here it says that the small divisions on a stage micrometer scale are 0.1 millimeters, but we have to convert them to micrometers. So what do is we multiply 0.1 by 1000, giving us 100 micrometer for each division. If we know that one stage micrometer division is 100 micrometers, then each division of the 40 on the eyepiece graticule is going to have, what we do is now we divide it by 100, and 100 divided by 40 is now going to be 2.5 micrometers for each eyepiece graticule division. So one division on the eyepiece graticule is going to be 2.5 micrometers. Now, coming back to our question here, it's asking what is the actual length of the nucleus in a plant cell. Now, we must first identify where's the nucleus. Here is the nucleus. And we could see that the nucleus is going to be 9 to 10 divisions of the eyepiece graticule. And we know that one division on the eyepiece graticule is 2.5 micrometers. Then if we multiply 2.5 by 10, the number of units on the eyepiece graticule here is going to give us 25 micrometers. So the length of the nucleus of the plant cell is going to be 25 micrometers. Question number 3. Insulin is a hormone synthesized from 2 polypeptide chains inside the cells of the pancreas. Which row shows the cell structures that are used in producing insulin and releasing it from these cells? So first of all, insulin is a hormone. And we know that hormones are made of proteins. And now we must identify the structures that are involved in protein synthesis. First of all, obviously the rough endoplasmic reticulum is surely involved in protein synthesis because they contain the ribosomes, which the mRNA code binds to for the polypeptide chain to be synthesized during translation. Therefore, after translation in the rough endoplasmic reticulum, these polypeptides are packaged into vesicles, which indeed move to the Golgi body for modification and packaging. Therefore, here we have the Golgi body involved, and the rough endoplasmic reticulum also involved. Now, are lysosomes involved? No, they're surely not, because lysosomes contain hydrolytic enzymes, and hydrolytic enzymes have nothing to do with insulin. Therefore, the only correct answer must be C. Question number 4. The electron micrograph shows a cell structure in a eukaryotic cell. Which statements about the cell structure are correct? First of all, we must even identify what the cell structure is in the first place. Here we could clearly see 9 microtubule triplets. So 9 microtubule triplets. And this cell structure corresponds to a centriole. So we now know that we are looking at centrioles, and this is what it looks under an electron microscope. Now, as we know that centrioles are made of microtubules, and microtubules are made of polymerized tubulin monomers, alpha and beta tubulin, which are indeed proteins. Therefore, centrioles are made of proteins. Now let's see the suggestions that we have here. 1. ATP is synthesized in this cell structure. As we said, these are proteins, and this is a centriole. It's not the mitochondria or the chloroplast. Therefore, ATP is not synthesized in this structure. For 2, the cell structure is made of protein molecules. As we said, it's made of alpha and beta tubulin. Surely it's made of protein molecules. 3. The cell structure replicates during interphase of the cell cycle. As we all know, this is true. It does replicate during interphase directly behind the S phase. Therefore, the answer is going to be C. Question number 5. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. The appearances were listed by letter. Which student correctly matched the numbered function with the appearance of the cell structure? Now, we must first identify what cell structures we are looking at. So for 1, synthesis of polypeptides. As we all know, what synthesizes polypeptides are going to be ribosomes on rough endoplasmic reticulum. Now, 2. Synthesis of lipids. As we all know, what synthesizes lipids and steroids such as cholesterol is going to be the smooth endoplasmic reticulum. 3. Packaging of hydrolytic enzymes that will remain in the cell. As we all know, after synthesis of polypeptides in the rough endoplasmic reticulum, they are packaged into vesicles and moved to the Golgi body for modification and packaging. So what packages and modifies these proteins is going to be the Golgi body. Now let's see the suggestions that we have here. For 1, we have either W or Z. Let's look at W. W, non-membrane bound spherical structures. As we said, that synthesis of polypeptides are going to be ribosomes, and ribosomes are non-membrane bound and they are spherical structures. Therefore, 1 is going to be W. For 2, for a smooth endoplasmic reticulum, we have either V or Z. Let's look at V, membranes which surround an enclosed inner cavity. So this is surely correct because an enclosed inner cavity means that it's a tubular structure. And as we all know, that smooth endoplasmic reticulum is a tubular structure too. Therefore, V is going to be correct. Now, for 3, we have either Z or Y. Let's look at Z. Membrane-bound sacs arranged as flattened stacks. As we all know, that Golgi body are made of many flattened sacs, arranged as a coin structure almost. Therefore, Z is going to be correct and the answer is going to be A. Number 6. The DNA of typical prokaryotes is naked and circular. Naked basically means that it's not bound or wrapped around proteins, such as histone proteins in chromosomes. And circular means it's basically circular, so it's not the linear structure that we have inside the nucleus. So which statements describe how the DNA of eukaryotes differs from the DNA of typical prokaryotes? So here we're trying to find the opposite of each definition, the opposite of naked and circular. Let's look at the suggestions that we have. A. Only DNA of eukaryotes has a nuclear envelope around it and a double helix. Both of these are not the opposite of naked and circular, so this is incorrect. B. Only DNA of eukaryotes has nuclear envelope around it and is circular. This is incorrect. The DNA of eukaryotes is not circular. And nuclear envelope is not the opposite of a naked. C. Only DNA of eukaryotes has proteins attached to it and is a double helix. This is incorrect. D. Only DNA of eukaryotes has proteins attached to it. So this is correct, opposite to naked and is linear, which is opposite to circular, so D is going to be the correct answer. Question number 7. A student carried out Benedict's test on 2 different types of milk, X and Y. A sample of each type of milk was heated to 100 degrees in a water bath with Benedict's solution, and the time taken for the first appearance of the color change was recorded. The results are shown in the table. Which row shows the biological molecule the student detected in each sample of milk and the sample of milk with the highest concentration of this biological molecule? So let's first start by identifying what is Benedict's test used for. It's used to test for the presence of reducing sugars. And a positive test would turn from blue to brick red. So the biological molecule present in each sample of milk must be a reducing sugar. And by the way, we do know that glucose is a reducing sugar, but because Benedict's solution is used to test for the reducing sugars in general, so we cannot guarantee that that reducing sugar is going to be specifically glucose. Therefore, glucose in this case is incorrect, and we could just say it's a reducing sugar. Sample of milk with the highest concentration of this biological molecule. So as we could see, the faster appearance of the color change was in X. That means that X had the highest concentration of reducing sugar because it had the fastest appearance. Therefore, the answer is going to be C. Question number 8. The diagram shows the structure of a monomer. Which molecules contain this monomer? So as we could see, the hydroxyl group is below the ring. This means that this is an alpha glucose monomer. Now, let's look for the molecules that does have this alpha glucose monomer. Here we have amylopectin. Yes, amylopectin is a polymer formed by the condensation of alpha glucose molecules, so this is correct. For sucrose, sucrose is formed by alpha glucose combining with beta fructose in a condensation reaction, so this is also correct. And for glycogen, is also formed by the condensation of alpha glucose monomers. Therefore, the answer is going to be B. 9. Which molecules are formed by condensation which involves a carboxyl group? So, first of all, we must identify what type of molecules might contain a carboxyl group. Now, here we have proteins. If we draw the structure of an amino acid, it would look something like this, with a carboxylic acid group on this side and with an amine group on the opposite side, and an R group or a side chain and a hydrogen atom. Now, as you can see here, the amino acid must always contain a carboxyl group. Therefore, all proteins must also have carboxyl groups. Now we are looking for proteins. Here we have collagen, beta globin, which is also a polypeptide, and collagen and beta globin. Now, the second molecule that we have here that must also contain a carboxyl group are triglycerides, because as we said that triglycerides are formed by 3 fatty acids which have a carboxyl group and glycerol, a glycerol molecule, which is an alcohol. Therefore, triglycerides and proteins must have carboxyl groups, and the answer is going to be D. Number 10. Which row correctly matches the function with the structural feature of cellulose, collagen, glycogen, or triglyceride? Let's start with A, and with cellulose. Now, we have cellulose here. The function is support. This is correct. It supports plant cells. So this is correct. And it is a fibrous molecule because it has a structural role, as we just said. Now, molecules held together by hydrogen bonds, this is also correct. The reason for this is because each beta-glucose monomer is rotated 180 degrees to each other, and this helps the formation of hydrogen bonds between the cellulose molecule in the polymer and between different or parallel polymers too. And branched chains, cellulose is not branched, so this is correct. Now, for triglyceride, it is an energy source because they have a high proportion of hydrogen to carbon bonds, and these release higher energy because there's a higher number of these bonds here. Therefore, this is correct. Fibrous, we know that triglycerides are not fibrous. Molecules held together by hydrogen bonds, this is incorrect. Actually, this is a reason why they are non-polar, because if they had many hydrogen bonds, they would be polar molecules. Branched chains, we know they are not branched. So this must be correct. Let's see the rest. B. Collagen. Function is strengthening. This is correct. It has a structural role. Now, it's a fibrous. This is correct. Molecules held together by hydrogen bonds. Actually, they are held together by hydrogen bonds and some covalent bonds in a triple helix structure. And they are not branched, so this is correct. Here for cellulose, we have support, fibrous. They are fibrous. Molecules held together by hydrogen bonds. This is correct. Incorrect, because we just said that they are actually indeed held together by hydrogen bonds because beta glucose monomers are rotated 180 degrees to each other. So this is incorrect. C. Collagen, strengthening, fibrous. Molecules held together by hydrogen bonds and branched chains. Collagen are not branched, so this is incorrect. D. Glycogen, it is a storage polysaccharide. This is correct. Fibrous, yeah, this is correct. It's not a fibrous because it has metabolic roles. Molecules held together by hydrogen bonds. This is incorrect. And glycogen is branched, so this is correct. So in general, A is going to be the correct answer. Question number 11. The diagram shows the amino acids in a polypeptide. An enzyme catalyzes the hydrolysis of any peptide bond between a non-polar amino acid and any polar amino acid. So here we have all the polar ones, and this is the only non-polar ones. How many small peptides and single amino acids will be formed by the action of this enzyme? Now we must look for amino acids attached to polar and non-polar at the same time. Therefore, there's going to be a hydrolysis reaction here and here. Here there's a single amino acid formed. Same with here. Now, let's count how many polypeptide chains have been formed. Here we have 1, 2, 3. And 13. We have 13 polypeptides chain. And now we're looking for the single amino acids. Here we have 1, 2, 3, and 4. Therefore, the correct answer must be D. Question number 12. Which properties of water reduce temperature changes inside the cells? Now, let's start with cohesion. This is not true because cohesion is the hydrogen bonding between water molecules, and this has nothing about reducing temperature changes inside these cells. 2. Latent heat of vaporization. For those who don't know, latent heat of vaporization is the energy required for water to evaporate. Now, as we all know, that water did not evaporate inside of cells, therefore this is incorrect. This is not needed, and this does not contribute to reducing temperature changes inside cells. 3. Specific heat capacity. For those who don't know what specific heat capacity is, it's the energy required to increase 1 kilogram of water, the temperature of 1 kilogram of water by 1 degrees. And as we all know, that water actually has a high specific heat capacity, meaning that it requires more energy in order to change temperature. Therefore, specific heat capacity is the only property that actually reduces temperature changes inside cells. Therefore, the answer is going to be D. Question number 13. Which enzymes is extracellular? Extracellular enzymes are enzymes that operate outside the cell. Now, let's see the suggestions that we have here. A. Amylase in saliva is an enzyme that catalyzes the breakdown of starch in the mouth. As we all know, that amylase is an extracellular enzyme because it has been released into the saliva or extracellular fluid. Therefore, A is the correct answer. Question number 14. The graph shows how the concentration of components of an enzyme catalyzed reaction changes with time. Which line represents enzyme with empty active sites? Now, please focus with me on this question. First of all, here we could see that line C, there's something, its concentration is decreasing. As we all know, as the reaction proceeds, the enzyme converts substrate into products. Therefore, C is going to be the substrate because it is decreasing by time. And here in A, we can see that something is increasing. Obviously, this is going to be the product. And for D, as we all know, as the reaction proceeds, there are more enzyme substrate complexes in the middle. More enzyme substrate complexes taking place. Therefore, D is going to be enzyme substrate complex. And for B, we could see something here decreasing, then going back to normal again. As we all know that when the number of enzyme substrate complexes are the most, the number of enzymes with empty active sites are the least. Therefore, B must be the number of enzymes or the concentration of enzymes with empty active sites, because it decreases here at the peak of the reaction and then increases again. Because the number of empty active sites at the beginning is going to be the most and at the end is going to be the most. Therefore, the answer is going to be B. 15. Which effects can non-competitive inhibitors have on enzyme-controlled reactions? First of all, we must start by asking, what are non-competitive inhibitors? As we all know, non-competitive inhibitors binds to an allosteric site on the enzyme, changing the shape of the active site. So it binds to an allosteric site, so any other site other than the active sites, changing the shape of the active sites. Now, let's see the suggestions that we have here. 1. Lower the Km value. Here the Km value is Michaelis-Menten constant, and it represents the affinity. And they are inversely proportional. So the higher the Km value, the lower the affinity. And the lower the Km value, the higher the affinity. So first of all, affinity is only impacted when it is a competitive inhibitor, not a non-competitive. Why is that? Because competitive inhibitors actually bind to the active site of the enzyme. So it competes with the substrate molecule. Therefore, it decreases the affinity. Only competitive inhibitors impact the affinity. And on the other hand, this is also cannot be correct for a competitive inhibitor. Why, you would ask? Because here it says to lower the Km value. As we said, lowering the Km value means the affinity increases, but this is not correct in case of a competitive inhibitor. As we all know, in a competitive inhibitor, actually the affinity for the enzyme for the substrate actually decreases. So the Km value must even increase, not decrease. Therefore, this is totally incorrect. 2. Reduce the concentration of the product. As we all know here, for a non-competitive inhibitor, the active site has been altered. Therefore, this would reduce the concentration of product because less substrate is actually being catalyzed by the enzyme. So this is correct. 3. Reduce the rate of a metabolic pathway. As we said, because the structure of the active site has been altered, then the rate of the reaction will also decrease. Therefore, 3 is also correct, and the answer is going to be D. 16. Which processes can allow transport into or out of a cell? 1. Active transport. Active transport is the movement of molecules against concentration gradient. This means it's an active process requiring ATP. 4.2. Facilitated diffusion. As we all know, facilitated diffusion, the molecules move down concentration gradient, but through a transport protein, could be a carrier or a channel protein. So through a transport protein, as we said, it's a down concentration gradient, so it is a passive process. It does not require ATP. 3. Osmosis is the movement of water molecules down water potential gradient. So all 3 of them has movement involved. Therefore, the answer is going to be A. Question number 17. Plant cells were put into one of three different concentrations of sugar solution, 10%, 5, and 2.5, with 10 being the highest and 2.5 being the lowest. The cells were left for 50 minutes and then observed using a light microscope. Which statements are correct? Cell Y had a lower water potential than the sugar solution it was put into. So let's look at cell Y. Cell Y here we could observe that it is a turgid cell. Means that there was a net movement of water into the cell. This is correct because here it says, it assumes that cell Y had a lower water potential, and as we know, that water moves down water potential gradient from higher water potential to a lower water potential, so this must be correct. 2. Cell Z was put into a 10% sugar solution. As we see here, the 10% sugar solution is the highest percentage of sugar. Therefore, in cell Z, we could observe that the cell is plasmolyzed. This means that there was a net movement of water outside the cell. Why is that? Because it has been placed in the highest concentration of sugar solution. This means that inside the cell was a higher water potential and outside was a lower water potential due to the high concentration of sugar. Therefore, there was a net movement of water by osmosis down water potential gradient outside the cell. So this is correct. Question number 3. Cell Z had a less negative water potential than the sugar solution it was put into. Don't be fooled. Less negative water potential means a higher water potential. Now, let's look at cell Z. Here it's the same cell. We said that it has a higher water potential than the solution it was put into. This is correct because, as we said, that water moves down water potential gradient. So, if cell Z had a higher water potential gradient and the outside had a lower water potential gradient, also there will be a net movement of water outside the cell. Therefore, it would become plasmolyzed and it would look like this. Therefore, all three of them is correct and the answer is going to be A. Question number 18. During telophase of mitosis, a scientist stains the chromosomes of a diploid animal cell with a fluorescent dye to observe the telomeres. This cell has 22 chromosomes. How many telomeres will the scientist observe in one of the nuclei? So let's just break down the question. We have to first know the difference between chromosomes and chromatids. So chromosomes have 2 chromatids. So chromosomes have 2 chromatids, which you could see here. There's the first one and the second chromatid. And each chromatid contains 2 telomeres. And for a single chromatid, it just looks like that and it has 2 telomeres on both sides. Now, here it says during telophase of mitosis. And as we know, during mitosis, what happens is that chromosome is split and each pair goes into the opposite sides. It says here, how many telomeres would the scientist observe in one of the nuclei? As we all know that each chromosome splits into sister chromatids and each of this pair goes to the opposite side of the cell, which would look something like this during telophase. So now we have 1 pair in each nuclei. Then this means that we have 22 now chromatids on each side. So here we have 22 chromatids, not chromosomes, okay, on each side. And here we also have 22 chromatids. And as we said that each chromatid has 2 telomeres, 1 on each side. So in order to calculate the number of telomeres, we must multiply 22 by 2, giving us 44, and the answer is going to be B. Question number 19. Methotrexate is a drug used to treat cancer. It can act as an enzyme inhibitor preventing synthesis of nucleotides containing thymine. Cells treated with methotrexate are not able to complete the cell cycle. A cell entering telophase is treated with this drug. Which stage of the cell cycle will be affected by the action of methotrexate? As mentioned here, it prevents the synthesis of nucleotides containing thymine. And as we all know that cell cycle consists of mitosis, and interphase. And we also know that during interphase, DNA replication takes place during the S phase of interphase. And we all know that DNA replication requires the synthesis of nucleotides to be able to synthesize new DNA by semi-conservative replication. So if we do not have nucleotides for DNA replication, then interphase cannot take place, so it will be impacted. Therefore, the answer is going to be B. Question number 20. Which stage of mitosis is correctly described? A. In anaphase, chromosomes line up across the equator. This is incorrect. This happens during metaphase. B. In metaphase, the centrosomes replicate. This is incorrect. This happens during prophase. C. In prophase, chromatids move to opposite poles. This is incorrect. This happens in anaphase, where the sister chromatids move to opposite poles. D. In telophase, chromosomes uncoil to form chromosomes, and this is correct. 21. The diagram represents a nucleotide containing cytosine. Which statements about this nucleotide are correct? So as we all know, we have two types of nucleotides. The first type is the DNA nucleotide and RNA nucleotide. By the way, they are not the same, they are different. And first of all, we know that the sugar or the pentose sugar for the DNA nucleotide is actually deoxyribose, and the sugar for RNA or the pentose sugar is ribose. So they are different, be aware of this. Let's start with the suggestions. 1. The carbohydrate could be ribose. This is correct, it could be ribose. The reason for this is because it says that the nucleotide contains cytosine. So this is correct. This would be incorrect only in one case, if it said that the nucleotide contained thymine. Because as we all know that RNA nucleotides containing ribose do not contain thymine, they instead contain uracil. Therefore, this is correct. Number 2, the organic base contains nitrogen. Yes, because we say that these are nitrogenous bases, so they definitely contain nitrogen. 3, base pairing occurs with guanine. As we know that cytosine binds with guanine via 3 hydrogen bonds, so this is correct. 4, cytosine is a purine. This is incorrect because purines are adenine and guanine. And purines are double-ringed structures. So the only double-ringed structures we have are adenine, as we said, and guanine. They are not cytosine. Cytosine is actually a pyrimidine with thymine. So 4 is incorrect, and the correct answer is going to be A. Question number 22. A section of DNA contains 73 base pairs. Which row represents the correct number of bases? So here we have strand number 1 and strand number 2. And here in strand number 1, we have 29 adenine. So here 1 and 2, we have 29 adenine. Therefore, in strand number 2, because they bind by complementary base pairing, is also going to have 29 of thymine, because adenine binds with 2 hydrogen bonds with thymine. So there's going to be 29 thymine in strand 2. And here in strand 2, we have 21 adenine. Therefore, also in strand 1, we're going to have 21 thymine. Now, as mentioned in the question here, it says there are 73 base pairs. Therefore, every strand is going to have 73 bases. Then, what we must do is we must add up 29 plus 14 plus 21, and then 73 minus this answer gives us 9. So V must be 9. Now, because we know that guanine is going to be 14 in strand number 1, then the cytosine in strand number 2 is also going to be 14. And then we follow the same method. We add 21 plus 14 plus 29. This gives us 64. Then 73 minus 64 gives us 9 also. Therefore, the only answer which fulfills this criteria is going to be A. 53. The photograph shows a section of a stem. Which labeled part is the xylem? Let's start by labeling the full thing. And let's start with A. A here we could clearly see parenchyma or packing tissue in these spaces. So parenchyma or packing tissue, let's just write parenchyma. For B, here for B it would just be lignified fibers, because this is what's surrounding the vascular bundle, so we could just call it lignified fibers. For C, here if you zoom in, you can clearly see larger circles. These larger circles are going to be the xylem. The smaller circles are the phloem. And for D, this is going to be the collenchyma, or as I call it, celery. Just remember it was celery. Celery stems have a hard part, and this is what's called the collenchyma. Therefore, the answer is going to be C. Question number 24. Some fungi cause wilting in crop plants by growing within the xylem vessel elements. Which processes will be directly affected by these fungi? So here we're talking about water transport. Now let's start with A, cohesion between water molecules. As mentioned here, wilting has happened in the xylem vessel elements. And wilting actually, this disrupts the cohesion column. So it is directly affected by the fungi. B, development of root pressure. Root pressure only is affected indirectly. The reason for this is that root pressure solely just depends on the concentration of solute in root hair cells. So therefore this is incorrect. C, mass flow during movement of assimilates. Here we're talking about assimilates, therefore it's about phloem sieve tube elements, not about the xylem, so this is incorrect. The uptake of water by root hair cells, we said that it will be affected but indirectly, because uptake of water also depends on other factors such as the water potential inside the root hair cells and the solute concentration too. Therefore, the answer is going to be A, because the cohesion pattern is the first thing that gets disrupted. Question number 25. Which features have a role in the transport of water in xylem vessel elements? 1. Capillary action. Capillary action is also known as cohesion tension. And cohesion is the hydrogen bonding between water molecules. And as we all know that hydrogen bonding between water molecules help keep the transport of water up the stem, because it helps maintain the transpiration stream and the transpiration pull upwards. Therefore, this is correct. So adhesion. Adhesion also has a role in the transport of water. The reason for this is because adhesion is the hydrogen bonding between water molecules and the cellulose cell wall due to its polar properties. This also helps maintain the transpiration column and stream and the transpiration pull upwards. So this is correct. 3. Hydrogen bonding. As we said that both of these depend on hydrogen bonding in the first place. Therefore, it's also going to be correct, and the answer is going to be A. 6. The loading of sucrose into companion cells involves a number of processes. Which process is active? First of all, let's recall the process of loading sucrose into companion cells. So let's start. Here we have companion cells and the phloem sieve tube elements. Let's label them. As we all know, the loading starts by companion cells pumping hydrogen ions to its cell wall by proton pumps, and this process requires ATP. So the pumping of hydrogen outside into its cell wall does require ATP. Then there is sucrose formed by the mesophyll cells. Then the hydrogen ions with sucrose both go back into the companion cell by something called a co-transporter protein. And this process is passive, so it does not require ATP. So once the sucrose is now inside the companion cell, there's a high concentration of sucrose inside the companion cell, it moves now into the phloem sieve tube element by diffusion, by normal diffusion, and this is also a passive process, so it does not require ATP. Then once the sucrose is inside the sieve tube elements, water moves into the sieve tube elements. By osmosis, down water potential gradient and a hydrostatic pressure builds up. Now, out of everything I just said, the only thing that is an active process that requires ATP is pumping of hydrogen ions outside the companion cell. So the only correct answer is going to be B. Question number 27. The statement describes events that occur during the movement of sucrose. Which sequence correctly describes what happened in a plant at a source during the movement of sucrose? So I explained and recalled the process of loading of sucrose in the last question. You can go and listen to it. So hopefully you've understood my explanation very easily and you're now familiar with the process of loading of sucrose. Now let's see the suggestions that we have here. We have either 3 or 4. So let's look at 3. Sucrose is loaded into a phloem vessel. This is correct because loading means that sucrose moves from the companion cell inside the phloem sieve tube elements. So this is correct. It is being loaded. Now let's look at 4. Sucrose is unloaded from the phloem vessel. First of all, unloading is the movement of sucrose from the phloem sieve tube elements into the sink cells. So into the cells of the sink, and this happens in the sink, not at the source. Therefore, 4 is incorrect. Now we have here 7 or 8. Let's look at 7. Water potential decreases in the phloem. This is correct because once the sucrose moved from the companion cells into the phloem, there's now a higher concentration of sucrose into the phloem. Then what happens is that water from adjacent cells moves into the phloem sieve tube elements, down water potential gradient, and then a hydrostatic pressure builds up. Therefore, 7 is going to be the correct answer for this. Now here we have 5 and 6. Let's look at 5. Water moves by osmosis into the phloem. You just mentioned this point, so this is going to be correct. And then 2, hydrostatic pressure is increased. This is correct because now there's so much water inside the phloem that now a hydrostatic pressure builds up, and then sucrose moves down that hydrostatic pressure gradient from a higher pressure into a lower pressure. So A is going to be the correct answer. Question number 28. Which are present in walls of capillaries? Endothelium. This is correct. Endothelium are present in walls of all blood vessels. So endothelium is definitely correct. And now here we have smooth muscle, elastic tissue, and collagen fibers. All 3 of them are not present in capillaries. They're actually present only in arteries and veins. Therefore, the correct answer must be A. Question number 29. The photomicrograph shows 3 white blood cells labeled X, Y, and Z. Which are correctly identifies these cells? Let's start with X. Here we could clearly see a bean-shaped nucleus. So a bean-shaped nucleus basically means that this is a macrophage or a monocyte. Therefore, X is a macrophage or a monocyte. Now, for cell Y, here we could see a granulated nucleus. A granular nucleus basically means that this is a neutrophil. So neutrophil is correct. For Z, we could see as a nucleus that takes over the whole cell almost, and a cell that is way smaller than the others. This is definitely the lymphocyte, because lymphocyte must have a really large nucleus because it has a higher rate of transcription and translation. Therefore, the answer is going to be C. And also, lymphocytes are the smallest of the white blood cells. Number 30. Which sequence of letters correctly identifies the order of events during the cardiac cycle? So the cardiac cycle first starts with the sinoatrial node initiating an excitation wave. The sinoatrial node is found in the right atrium, and it initiates an excitation wave. So 1 is going to be X. Then after that wave spreads throughout the atrium, the atrial wall contracts. So 2 is going to be T. Then after that happens, here the excitation wave finally reaches the atrioventricular node, which acts as a relay and delays the impulse at a fraction of 0.1 seconds It delays the impulse. The reason for this is so the atria and the ventricles do not contract at the same time. So 3 is going to be V, and 4 is now going to be also U, because it is delayed at a fraction of 0.1 seconds. Then, after this delay happens, the wave now travels at the base of the ventricles to something called Purkinje fibers, found at the base of the ventricles. Once the wave reaches the Purkinje fibers, so Purkinje tissue is going to be 5, now the ventricles are able to contract. So Y is going to be 6. And the answer is going to be A. Question number 31. The graph shows the effect of 3 different partial pressures of carbon dioxide on the oxygen dissociation curves for human hemoglobin. Which effect does decreasing the partial pressures of carbon dioxide, so decreasing the partial pressure of carbon dioxide, have on hemoglobin? As you can clearly see, decreasing the pressure of carbon dioxide makes the graph shift to the left, and increasing the pressure makes it shift to the right. Shifting to the right means that the percentage of saturation is actually going to be lower at the same partial pressure of oxygen. So now let's see what the increased concentration of carbon dioxide actually does to the red blood cells and the saturation of hemoglobin. Now, when carbon dioxide concentration increases, now more carbon dioxide combines with water to form carbonic acid. This is obviously catalyzed by the enzyme carbonic anhydrase. Then this carbonic acid dissociates into hydrogen ions and bicarbonate ions. The hydrogen ion stays in the red blood cell and the bicarbonate leaves the red blood cell. Now what happens is now we have so much hydrogen ions. What happens is these hydrogen ions now combine with hemoglobin, forming hemoglobinic acid. And oxygen is now released. The reason for this is that hemoglobinic acid has a very low affinity for oxygen, so low affinity. Therefore, it releases oxygen much easily. Now, here we're looking at the effects of decreasing the partial pressure of carbon dioxide. So decreasing the partial pressure of carbon dioxide means that the affinity increases. Therefore, it's going to become more efficient at taking up oxygen in opposite to hemoglobinic acid. And because, as we said, that the hydrogen ions are the reason why oxygen is released in the first place, then when the concentration of hydrogen ions decreases, this means that oxygen is no longer released. Therefore, it becomes less efficient at releasing oxygen, and the answer is going to be C. Question number 32. What is correct about the transport of carbon dioxide by blood? 1. The enzyme carbonic anhydrase catalyzes the formation of carbonic acid in red blood cells. As we mentioned earlier, the enzyme carbonic anhydrase catalyzes the reaction when carbon dioxide combines with water, forming carbonic acid. Now this carbonic acid dissociates into hydrogen ions and bicarbonate ions. Now these bicarbonate ions leave the red blood cell, and carbon dioxide is now transported as bicarbonate ions in the plasma. So this is correct. 2. Carbon dioxide diffuses from active cells to red blood cells and reacts with water. As you said, it reacts with water by the enzyme carbonic anhydrase, which catalyzes this reaction. Therefore, this is correct. 3. Carbonic acid dissociates, forming hydrogen ions that combine with hemoglobin to form carbaminohemoglobin. So this is incorrect. Actually, carbaminohemoglobin is formed by carbon dioxide combining with hemoglobin in a terminal amine group. Now, here, when hemoglobin combines with hydrogen ions, it actually forms hemoglobinic acid, not carbaminohemoglobin. So this is incorrect, and the answer is going to be B. Question number 33. Which statements about all bronchioles are correct? 1. They have goblet cells. Now, this was a very common misconception. Bronchioles never have goblet cells, or a very, very small amount, but it's so insignificant. What has goblet cells are trachea and bronchus. So this is incorrect. 2. They have ciliated cells. This is correct, in order to sweep the dust and the mucus. 3. They have muscle tissue. This is correct. They have smooth muscle to be able to control the volume of air that actually enters. Therefore, the answer is going to be D. 34. The graph shows the volume of air breathed out quickly and with force, allowing a deep breath in for 3 different people, X, Y, and Z. What is a possible explanation for the differences in the volume of air breathed out in these people shown? Obviously, I know you're very hardworking students and you know what is chronic bronchitis and emphysema. So let's start with the easiest, and it's emphysema. Emphysema, as we know, is a decrease in surface area of the lungs because the alveoli actually bursts. Then, what happens when the alveoli bursts? This means that less volume of air, a significantly less volume of air is breathed out by people with emphysema. Therefore, Z is going to be emphysema. And now we have normal lung function. We know that normal lung function is going to breathe out the most volume of air. Therefore, normal lung function is definitely going to be X. And therefore, the answer is going to be C. Question number 35. The diagram shows some of the pathogens that cause disease in humans and some of the ways they are transmitted. What is the correct pathogen and method of transmission for the disease tuberculosis? So the pathogen of tuberculosis is Mycobacterium tuberculosis. And it is a bacterium, so it is a bacterium that's transmitted by airborne droplets. So airborne droplets is also coughs and sneezes. Therefore, the answer is going to be B. 36. The proportion of local population who have malaria in area R is higher than the proportion in area S. Which factor causes this difference? A. Area R has a more humid climate than area S. This is actually correct. The reason for this is the insect vector for Plasmodium, or the pathogen that's causing malaria, is female Anopheles mosquitoes. And as we all know, that Anopheles always favor humid climates. Therefore, an increase in the insect vector makes it more likely that malaria is transmitted even more. And more people have malaria in the area that has a higher proportion of female Anopheles. Therefore, A is correct. Let's see the rest. Area R is nearer to the equator than area S. Nearer to the equator is actually a really vague answer, because near the equator might mean it's too hot, might mean it's too cold, might mean it's humid. So it's a really vague answer, therefore this is incorrect. C. There is a higher population in area R than S. Here, as we said, the insect vector is female Anopheles, so it has nothing to do with the population of humans. It has something to do with the population of the Anopheles itself. Therefore, this is incorrect. D. There's less sewage treatment. Okay, sewage treatment, you know that cholera is what is transmitted by fecal-oral route, so this would only impact cholera, not malaria. Therefore, this is incorrect, and the answer is going to be A. Question number 37. Species X is a single-celled eukaryote. Species X has been genetically modified to produce penicillin, which does not harm the cell walls of species X. What may be concluded from this information? First of all, penicillin prevents the growth of bacteria's cell walls. The reason for this is because it inhibits the enzyme that causes cross-links between them. Therefore, let's see the suggestions that we have here. The cell walls of species X are chemically different from those of bacteria. This is correct. This is why it's not impacted by penicillin. 2. The cell walls of species X are made of peptidoglycan. This cannot be correct because, as we know, that penicillin actually acts on peptidoglycan, stopping cross-links forming between these molecules. and then the bacteria bursts. The cell walls of species X is made of cellulose. Here we cannot guarantee that's actually going to be cellulose. The reason for this is it says it has been genetically modified, so it might be anything, not necessarily cellulose. So the only correct answer is going to be B. Question number 38. When exposed to an antigen for a second time, memory cells stimulate a secondary immune response. Which correctly shows the secondary immune response? First of all, in the primary immune response, we know that memory cells are produced. So, a feature of secondary immune response is that memory cells are already produced from the first primary immune response. Therefore, what happens is when this memory cell encounters the antigen for the second time, it divides at a much higher rate into both plasma cells and more memory cells. Therefore, the plasma cells secrete antibodies specific for this antigen. Therefore, the only correct answer must be C. And A is incorrect because memory cells do not itself secrete antibodies, it first has to divide it into plasma cells. And also D is incorrect because here it's assuming that the memory cells also secretes antibodies, which is incorrect, and divides into memory cells. So divides into memory cells is correct, but what secretes the antibodies is plasma cells. And B is incorrect because the memory cell doesn't just divide into plasma cells, it also divides into more memory cells just in case this antigen is encountered again. Therefore, C is going to be correct. Question number 39. Monoclonal antibodies are produced for use in diagnosis or treatment of disease. To obtain the antibodies for an antigen, a mouse is injected with the antigen. Some of the events in the production of the monoclonal antibody are listed. What is the sequence of the first 4 events in the production of monoclonal antibody? So first of all, the antigen is actually injected into the mammal for a primary immune response to take place. Therefore, we have a choice between 3 and 5. 3 is the B lymphocytes that recognize the antigen, multiply and become plasma cells, and plasma cells are removed from the mouse spleen. So 5 is totally incorrect because first, the B lymphocytes actually recognize the antigen that has been injected and multiply and become plasma cells, which secretes antibodies. Now, here we have 5 plasma cells are removed from the mouse spleen. This is correct. And we also have 1. Plasma cells are then fused with cancer cells to form a hybridoma. So plasma cells fuses with cancer cells, also known as myeloma cells, by something called a fusogen, forming the hybridoma cells. The reason why this fusion occurs is because plasma cells secrete antibodies, but they do not replicate very quickly. But myeloma cells or cancer cells divide very quickly, so fusing them together forms the ideal hybridoma cells. Now, we have a choice between 2 or 4. 2. Hybridomas that secrete the required antibodies are identified and cloned, and hybridomas divide by mitosis and secrete antibodies. Now, 2 is definitely incorrect because hybridomas that secrete required antibodies are identified and cloned. The hybridoma anyways means it's already cloned because it has been fused with myeloma cells, so this is incorrect. What happens is the hybridomas divide by mitosis and secrete their required antibodies, but there's no such thing as something is cloned. Question number 40. Which row describes passive immunity? So passive immunity is immunity obtained without an immune response being generated, such as injecting the body with antibodies. So antibodies, do not get confused with antigens. Injecting the body with antibodies does not generate an immune response. Now, let's see the suggestions that we have here. Triggered by an antigen. As we said, it has nothing to do with antigens. If it was triggered by an antigen, it will generate an immune response and it will be no longer passive immunity. So this is correct. Involves an immune response. We said it doesn't, so this is correct. Memory cells produced. Because an immune response has not been generated, then surely memory cells will not be produced, so this is correct. And permanent protection does not take place. The reason for this is also because memory cells are not produced, so the answer is going to be D. Thank you very much for watching this video. I'd highly appreciate it if you would take a moment out of your very busy day and subscribe to this channel and like my video. Thank you very much.